theme from all of our panelists that timing in a pandemic is key, and that there is really a coordination needed in a public-private partnership. Fortunately, we do this every year for seasonal flu, so we're well practiced. We know that we have the relationships, we know what's expected, but every pandemic strain will be unique and will create its own problems, as we heard some of them during 2009. So I think we've touched on most of the different areas. So I'm going to just give everybody a quick question and then give the audience here some time to think of some additional questions for the panelists, and I'm sure there's going to be many. So Wenjing, back to you. You described the GISRIS and the surveillance system and the partnership with industry very well for our seasonal capacity that happens twice a year. During 2009, what were some particular challenges? And I know Bruce mentioned the switch, which is what a group of experts have been working on for a number of years now, identifying several bottlenecks that occurred in 2009. And it's an ongoing process of how we might take each of those bottlenecks and come up with solutions. But so if you could weave that into your response, that would be great.
So while we could learn a lot from 2009, but I don't think 2009 is a perfect example to reflect on the development of vaccines, because there are quite a lot of aspects did work well. So I give you some example. Well, one is the virus detection. So when we are talking about the vaccine development, we always need to bear into mind that there is a time lag between the first case and the actual pandemic detected. I didn't count the days in 2009, but a later example is 2013, the H7N9 outbreak in China, where the first case in February, and the IHR notification of the case, it took 40 days. So in 2009, the network, the GISRIS, because of the past dozens of years building, the capacity is there. The virus detection was smooth. The surveillance was sufficient in a way that viruses being shared, GSD were shared efficiently. So the timing, another things we were lucky or worked out well is the timing. The pandemic was detected in April. This was the time slot that the vaccine production for the Southern Hemisphere already finished, and the vaccine production for the Northern Hemisphere not yet started. So we were not in a situation in the middle of the seasonal vaccine production. So there's not an issue of the switch, whether we should stop now of seasonal vaccine production to start the production of pandemic of vaccine or not. So we were luckily not in this type of dilemma in 2009. Another example is the reassortment of the CVV, the candidate vaccine virus. It was so smooth. The H1N1 pandemic virus worked so well in the hands of the WHO collaborating centers to develop the candidate vaccine virus. And also, it worked out so well. The yield of this H1N1 was actually better than average of the seasonal vaccine virus yield. So again, I could also add from the surveillance, there's also enough evidence provided when there was a time to decide which are the risk group that should receive vaccine in the first round. There was enough surveillance information available. So all of these actually were worked on well, so smoothly. It's a little bit different from the scenario that was in our mind, the pandemic preparedness based on H5N1 scenario. But there's also a lot of challenges were encountered in 2009. First is those fixed response. So the pandemic preparedness plan in 2009 is that the response is fixed or bind to the specific phases. Once in a pandemic, there's a list of actions will have to be taking place. So this is written in many national response plans. Another challenge we encountered at that time is the biocontainment recommendations. So many of you may remember that the virus actually is circulating already in the community and causing actually mild disease. But for the vaccine development, still there's requirement of BSL-2 plus was this type of containment requirement, which was not existing in many manufacturing plants at that time. Well, and the third, already mentioned by my colleagues, is regards was the regulatory capacity. In those countries that are receiving vaccines, donated vaccines, there's not such a mechanism in place with regards whether or how to receive or not. Also, with regards to deployment, aside from those legal aspects, there's also policy aspects. For example, when the pandemic already towards the end, there's already other seasonal virus circulating. Still, people are using monovalent H1N1 vaccine. So this was what happened, I think, a lesson we should learn. So this is type of challenges we had in 2009.
Okay, thanks. So, Karen, you mentioned that development of improved potency assays is an ongoing thing. Could you give us just a snapshot of where we stand with that? Because it is a key time sink, I think, for pandemic vaccine development.
Yeah, sure. Actually, BARDA stood up the influenza vaccine improvement plan, which CDC, CBER, and NIH all participated in together with BARDA, and that was one of the things that clearly fell into the CBER having the lead in the regulatory domain. And there was a consortium that was set up that includes other essential regulatory laboratories, also manufacturers trying to test different assays. And there's been a lot of them, actually. Jerry Ware published a very nice article. I have the reference there on my desk, but it describes all the different methods that have been tested in a collaborative way. And there are a couple, I want to say ELISA is one of them, that really seem to have potential, but there's still questions about how quickly could you develop the appropriate monoclonal antibodies that would be needed for that. And there's another one, I just can't remember off the top of my head which it is. So there is a lot of progress that's been made, but it's still a work in progress in terms of really figuring out would one of these be more suitable and really fulfill the criteria of having rapid reagents or maybe even pre-existing reagents available rather than having to develop strain-specific anti-sera once you have once you're manufacturing the virus. I think the other point that Jerry made in the article, which is really well taken, is that one other thing that's happening is we're having a lot of different vaccines being developed, and it's really a question, these really, the approach that was taken is to ask what kind of a potency assay could replace the existing potency assay for the traditional inactivated influenza vaccine. And now that we're having other, different kinds of, vaccines in the pipeline, is it really, even feasible going forward to think that you're going to have just one potency assay that will be the right assay for all? So it's a dynamic issue. My hope is that we can get to a place where there can be a better potency assay for the more traditional live inactivated influenza virus vaccines. But I think as different vaccines become available, that some of these things will have to be considered, and it may not be one size fits all.
Yeah, which creates a whole new challenge. Yeah. Clem, I was interested to hear that the fill and finish capacity is not available here. Is there a warm base for? I'm taking it's outside of the US that the fill and finish. A lot of fill and finish would occur if we had to fill and finish 600 or 700 million doses.
No, I think actually not exactly, because I think what I was referring to is, and I think this is actually something that applies even to when you hear new technologies, is manufacturing capacity, installed manufacturing capacity is what's required. So If you look at, let's say, cell culture, recombinant, which have been around, recombinant's about 5 years old, capacities maybe in the millions of doses, not tens of millions. And cell culture, it's taken about 10 years to get to 20, 30 million. It takes a while. So if you take the same thing with fill finish, the companies today, first of all, they tend to fill in pre-filled syringes now because that's what the market wants. The companies' fill finish strategy is based on what they sell today. So they need capacity of 150, 160 million doses. What I'm talking about is U.S. Capacity. Not all of that's filled in the U.S. Primarily pre-filled syringes or multi-dose vials. Now, in the event of a pandemic, you would need, say, 700 million, and you would need to fill probably multi-dose vials. I think that, wouldn't be us deciding, but BARDA and others would tell us what they wanted. In order to do that, it's not that the capacity doesn't exist in the U.S., it's that the capacity needs to be found. You need to do a tech transfer so that you can actually use that capacity when needed, and it has to be licensed by the FDA. So all of that has to be thought of now to say, okay, we can produce the bulk, but then how do we fill it as quickly as possible so that the filling doesn't become a bottleneck? And that would involve going to what are known as contract manufacturing organizations that do fill finish, but then you would probably have to pay them a reservation fee and also invest in the tech transfer. So it's Murphy's Law or the devil's in the details. There's always something.
Right. We recently went through a pandemic exercise at CDC, and the one working closely with BARDA, and the glaring gap was syringes. That even if we had the vaccine capacity, we didn't have the syringes to distribute.
Yeah, that's another, those are all of these things. And that's not to say that the capacity isn't in the U.S., but we, as manufacturers, we plan our supply chain for what we make for seasonal. Pandemic is a whole new ballgame, and we are thinking about it. We're talking to BARDA. There have been various initiatives to try and improve that, and we're better off than we were, but we still have a ways to go.
I don't want to hog the whole question time, but just a quick question for Bruce. You mentioned some of the unexpected things that came up with respect to shipping and distribution, particular permits, fumigation, things like that were needed, that were unexpected. Is anybody gathering a list of those to address them next time round?
I think that we felt like it was like for some improv skit about the number of things you could come up with that honestly the next day it was like, who invented this one? And the volcano in Iceland probably topped it off, which probably some of you were stranded in places where you either wanted to be or not wanted to be. But so I think in a way this was a first attempt, and it felt like an exercise, but real fire exercise, and I hope that we can learn everything from it. I guess the one piece I would say is that, the people doing this, including me, come up through medicine and public health, not logistics. And maybe the people who do this for a living should be, closer to this because they do that part every day.
Good point. Okay, and finally, quickly, Steve, I didn't quite understand. You talked about pre-qualification and then eligibility assessment of recipients for vaccine donations. What does that eligibility entail exactly?
Yeah, the pre-qualification is WHO's pre-qualification because many of our member states rely on that. They don't have their own regulatory authorities. So but for each of the vaccines, we have to do that. Sometimes it goes very quickly. It can take as little as 1 day, but for other manufacturers, it could take up to 20 days. The eligibility is, looking at the 194 member states, obviously not all need donated vaccine, but the question of who does is really the eligibility question. And those two criteria are, do they have domestic production? Well, that's easy. There's very little domestic production outside of a very limited number of countries. And two, do they have the capacity to buy it on the commercial market? Well, there the answer is almost always no, because they don't have pre-contracts for it, and much of the supply has been pre-contracted. Getting that commitment for 10%, which is at the heart of the PIP benefit sharing scheme, getting that commitment for the 10% as it rolls off the line from the manufacturers is absolutely key. The other thing that's key, and Professor Fiedler raised this point yesterday, and it's an untested issue with PIP, is getting countries to say they will release that, especially if it's a severe pandemic. And so that's a really tough issue, but it's very important. The U.S. did make that commitment at a presidential level to give the 10%. Stands as a good example, but that was in a different context. So that's the answer.
All right, so I'm sure there's lots of questions here in the audience. So yeah, please, I can't see everybody, so raise your hand or your name tag and just say who you are also. Thanks, Peter.
Hi, Peter Dashak, Chair of the Forum. I wanted to start off with Bruce and Steve, or whoever else wants to answer it. If something similar to H1N1 happened next year, how different would those two lines be? The virus versus the vaccine production. So a completely new reassortant virus. You've got to go from scratch, and it's moving rapidly. Are we really going to get to that point where we're beating the viral epidemic curve?
It's clearly not for Steve. It's really for Rick Bright, and I think that's where BARDA is looking at new technologies to try to collapse that time frame. Then there's the whole issue of the vaccine that you have before you need it, and the universal vaccine or whatever people are calling that. I will say that when Bill Gates demonstrated that model from the modelers, he did it at the Shattuck Lecture at Harvard, where he also used that opportunity to announce a grand challenge for a universal influenza vaccine, acknowledging that there's lots of stuff going on, all the things that NIH is doing in their strategy, a number of other countries in their public sector are doing that. But in addition to call for innovation, to have people who aren't part of that club to take a look at it to see what they could offer. They had hundreds of applications, and we'll learn, I think, early next year what that looks like. I think the idea was to think about things that could get vaccine firmly in front of a wave, rather than hoping you could, do it quickly enough. I think the technology, and I won't speak for Rick and all the work that BARDA is doing and all the companies are doing, are trying to compress that. The things that you heard Karen talked about are also trying to compress that. But I think the reality is that we should expect that we may not meet that first wave with where we are currently.
Steve, you want to comment? And then Clem.
Thanks. No, I also took Peter's question to be, are we ahead of, maybe doing better with that curve globally as well? And there I think the answer is the PIP framework puts us in a much better position. And Ann Huvos is going to speak about that. Because the PIP framework has pre-contracted for almost 400 million pandemic vaccine doses, and because we are agreeing these terms and conditions. So the recipient countries will be set to receive without having to go through the negotiations for that. But all the other deployment issues, all the other Murphy Law issues, whether there's a list being kept somewhere. I mean, this is worked on, but it requires resources to do that.
Clem, you want to?
I think we are better off than we were in 2009 because we have licensed adjuvants, we have recombinant and cell culture vaccines. We barely had cell culture at the time. Those do improve the time a little bit, but as Bruce said, if you want, or Dr. Bright would say, if you want to get a vaccine in 6 weeks, these technologies aren't going to do it. You need to find either a universal vaccine or potentially something like a DNA or mRNA vaccine. But all these technologies are unproven. That's not to say that they won't get there, but I think we're talking 10 to 20 years, and it'll take a while for that to come forward. So we have what we have today, and we all have to make the best of it. And that's this balance of not, I'd say throwing out the baby with the bathwater, is remembering that the technologies we have today will form the backbone without not investing enough in these new technologies that would really improve the curve.
Wenjing?
So just maybe from global perspective, different from 2009 is now that we have the severity assessment. So if it were now or next year, maybe if we just look at the disease severity, if the disease only causes, for example, the significant disease in pregnant women, maybe the vaccine production will set to a certain portion of H1N1 monovalent, while still continuing the seasonal vaccine. So this is one difference. The second is about national pandemic preparedness planning. So a whole set of national pandemic preparedness planning is being developed and reviewed. And that actually includes those regulatory side, those logistics deployment side. These are things are different from 2009.
I think there was a question over there. Yep.
Yeah, this is Matt Zahn with Orange County Public Health in California, representing IDSA. So I guess my question I have is for vaccination safety and effectiveness. You know, once you've made the decision, you know, and we've chosen the vaccine and putting shots into arms. Looking back to the 2009 event, were there a lesson learned from that event that would apply to how, you know, how this data is gathered and how the surveillance data is gathered at this point, and how would that look, you know, CDC versus, you know, versus private industry looking at effectiveness data? I don't know how much you could speak to that.
Yeah, thanks, Karen.
Yeah, you know, what FDA does when it licenses a vaccine is it actually does clinical trials that show effectiveness against a confirmed influenza disease. And that's what you'll typically see in the label. But you're correct that every year, though, the vaccine changes a little bit when new strains evolve. And so what typically is done there is that CDC has actually established some centers that every year look at vaccine effectiveness sort of on an ongoing basis. And maybe Jackie can talk about that. Are you familiar with that?
Sure. Yeah. So those would be in place. We would be monitoring the effectiveness, and there were many clinical studies, but what the CDC does with their US network is in a real world situation, evaluates how well the vaccine is working. And just to speak to your safety, CDC also has safety monitoring. Of course, there were some concerns with some pandemic vaccines that had adjuvant. We didn't have that experience in the US, but in Europe there was a concern. And again, that is a problem once you deploy vaccine to a large number of people. How do you monitor the safety? And fortunately, I mean, there were multiple studies that sort of were put in place, but almost after the fact, and it took a long time to really understand the safety issues with the adjuvanted vaccines.
Let me just speak to the safety piece of this one. I think Jackie covered the system that looks at effectiveness. So required reading from this panel and probably everyone in the room was Dr. Feinberg's 1976 manifesto about what happened in that pandemic, and we tried to learn everything we could from it. And maybe John Barry's book as well. But I think that the key lesson was, particularly given that experience in 1976, was having the most robust safety system we had and to be as transparent as possible. When you think about the backdrop of considerations about vaccine safety, much broader than flu, of where this took place. So we pulled out all the stops. We used every system that was available in the U.S. government, not just within HHS, but the Department of Defense. We supplemented many of those. We used immunization registries in a new way to try to expand that. And the other thing we did is on the transparency side, we had our National Vaccine Advisory Committee hold a monthly meeting to assess what the data looked like. There wasn't much. It took a long time to come in. And the ultimate point was it took about a year to figure out that there was an increased risk of 1 in a million of GBS, which is what we thought it might be, but at least you could demonstrate it. But wanted to not only have the best system, but a system in place where we could communicate that out to people who might worry. Because there was a lot of worry about this. And even though, as Karen said, this is making vaccine the same way we make it every year, the only thing that was different was the virus. Because it was a novel vaccine, it was perceived as a novel virus. Because it was a novel virus, it was perceived as a novel vaccine, and all the things that go along with that. So I think that served us well. We ramped that down. I think it could get ramped up again, but I think it built mostly on the systems that are already in place. That's in the U.S. That doesn't speak to what goes on globally, and that gets to Steve's point about when you if you start seeing adverse events, then how do you call them in terms of, is there a system that can identify causal versus temporal? And then if they're causal, what do you do about it? And is there some equivalent of a compensation program like the United States has or many other countries have, but there's not a global equivalent of that?
Thanks, Bruce. And I think there's another question over there.
Hi, Gabrielle Fitzgerald. I have a question for the WHO colleagues on the panel. So following the Ebola, West Africa Ebola outbreak, there was a lot of talk about how the new WHO Health Emergencies program was, really revamped and worked in a new way. I'm curious how the work that you're describing on pandemic influenza relates to the Health Emergencies program, and if all the lessons you've learned from this effort have been brought into the Health Emergencies program, or are they proceeding in parallel? Thanks.
Wenjing, you want to?
So I'll start.
Okay.
So, yes, so after Ebola, the WHO's health emergency program is being established and is being strengthened. The influenza program is within the WHO's emergency program. Well, the idea is that influenza has been or is a threat, a persistent or continuous threat for health. Although we are talking about influenza pandemic is every 10 years or every 15, 20 years. But every year, actually, there are seasonal epidemics occurring. So this is one. The second is that to prepare for pandemic, several people already mentioned, it is through seasonal capacity building, seasonal vaccines, to get familiar with. It is indeed an opportunity for influenza that may not be present for other disease, for other unknown emergencies. The third is with regards to platform. Anything cannot be built in a country stand alone. For example, just for influenza. In a country, it is a system, it's a whole platform, it's a whole health system. It is the same institution dealing with influenza, dealing with MERS, dealing with Ebola and unknown emergent ones. So it is the same platform of IHR, of JEE, post-JEE, preparedness, etc. But using influenza as the example to move forward from the common platform to the extreme that a country could strengthen the capacity. I think that has been the deliberation. So for influenza is being benefited from this common platform of health emergency. On the other hand, influenza also strengthened a common platform of health emergency to those unknown emergencies to the extreme possible.
Steve, do you want to comment?
Yeah, just very quickly, I guess I would say the lessons very much brought into the WHE program, but the issue is sometimes just lack of the tools and resources. So I put up the slide very quickly. You didn't have probably time to read it all on the IHR and things where more work was needed. And principally here we have, some financing mechanism for the IHR. It doesn't exist. Some compliance issues remain something that we have to work very hard on. And the issue, and this was mentioned yesterday, also the access to pathogens and their genetic sequence data, which is not seen as required under the IHR, is very important. But that means you're going to have to think about benefit sharing when you get access to those because of the Nagoya Protocol, which is going to come up more at the next panel.
KG, do you have a question?
Thanks, Jackie. I think by the time vaccine was available, it was already clear that the impact on mortality was relatively milder than everyone feared. And I think that colored everything. And I think in the entire process, the technical processes, in fact, worked really pretty well, as Wenjing mentioned. They went along pretty smoothly because it was an intensification of what was already in place. Everything after that was difficult because it was much more ad hoc and there was very deliberative and very difficult. If the pandemic had been severe, if we were seeing something on the order of an H5 or an H7 transform, do you think that the system would have come together and acted better and would have been more, decisions would have come more quickly? Or do you think it would have fragmented and in fact we would have seen a different set of behaviors where vaccines do not lead? Because I think that this is a very big issue.
Who would like to tackle that one? Bruce?
I'm no longer with the government. I'll take that one. No, so I think it's I think that there is maybe the urgency would have shortened some timelines. But I don't think they would have shortened them significantly. A lot of things were already collapsed, and it wasn't business as usual in the bureaucracy either. So I think there was an effort to try to coordinate, to try to collapse many of those things. I do think that the issue that you raised at the end is about sharing would be different. The decision from the United States was in September, when we had a clearer sense than we did in April about severity. But if you can imagine, I'll imagine that a different set of players might have called it differently, and that the ability for vaccine to be donated from a country, knowing that you're taking it away from the citizens of that country and giving it some other place, I think would be a lot more problematic. In this report, which is worth looking at from the deployment, the retrospective and the deployment, it also talked about pledges initially of both vaccine and dollars. And I guess this happens all the time, whereas what people pledge doesn't necessarily find its way to the bank. But there was a significant drop-off between what people pledged and what they gave. I'm sure there's lots of reasons for that, but I guess I would say that when the severity would be turned up significantly, I think there would be a difference in terms of willingness to move things to other places.
Okay, Wenjing, final word. I think we're going to close it here.
So I think, for 2009, if it were a severe pandemic like H1N1, I don't think, the situation would be better, because the response, the plan, everything was developed according to the H5N1 severe pandemic. But for the future pandemics, if a pandemic is a severe one, for some of the decisions may be simpler, because it's just go ahead, no seasonal, nothing, go ahead. But I think, this is quite difficult to say now. So what we've been doing now is really to develop certain principles and criteria with regards to make a decision or not. For example, the switch of vaccine production from season or not. And the communities communicate this in advance to the community, so that the community could understand when a decision made is always made based on the best possible evidence at that time to ensure the best possible public health outcome.
So I think we're going to close the panel here. I'm mindful of the time and everybody needing a quick break. I think we've highlighted a lot of the global challenges, particularly for distributing equitably vaccines to countries that may not have the infrastructure, the regulatory capacities, but clearly in any pandemic, and no doubt in a severe pandemic, would be in need of vaccine. And some of these things are being addressed by the PIP framework. I think we're going to hear, it's a really nice segue, we're going to move into the discussion after the break on how the PIP framework is responding to some of these challenges. So if everybody could join me in thanking our panelists, I think it's been a really